Hi, my name is Matthias Lienhard and I will now present my Python tool for long-read transcriptome sequencing analysis. With PacBio IsoSeq and Oxford Nanopore, there are several protocols for full-length transcriptome sequencing available. This technology avoids many shortcomings of the short-read RNA-seq, in particular to resolve transcript isoform level expression and alternative splicing analysis. However, to unravel the full potential of these experiments, specific tools are needed, and that's why I implemented the IsoTools Python package. The workflow typically starts with some pre-processing and alignment of the full-length transcript reads to a reference genome. The aligned reads then serve as an input for the methods I implemented in IsoTools. The first step of the analysis is the transcriptome reconstruction, by importing the reads and grouping them to transcripts, and the transcripts get grouped to genes. Further, transcripts are compared to known gene annotations to assign gene names. During this import, a number of QC metrics are computed to assess artifacts and biases. Transcripts not found in the reference annotation are classified according to the nature of novelty by biologically motivated categories, for example, novel exon skipping events. For each gene, all relevant information is stored, such as the list of transcripts, the number of long reads supporting each transcript, the quality control metrics, and so on. Further, the splicing information of the gene is encoded in a graph structure, which is used by downstream algorithms. The genes are integrated in an efficient tree-based data structure that allows fast access to the genes either by position or by name or ID. All these information can also be directly assessed by the user when using the Python API. IsoTools provides several statistical methods based on the gathered information, such as saturation analysis to judge the appropriate depth of sequencing. It can also compute 2D embeddings of different samples based on their alternative splicing and compute differential splicing between groups of samples. To export the relevant information, I implemented a flexible filter query syntax that facilitates the exact definition of what transcripts to export, and several output formats are supported. Last, there is a submodule providing functionality to visualize the data, for example, as sashimi plots. Now, in this talk, I will focus mainly on the graph structure and how it facilitates the differential splicing. What I'm using is called a segment graph, where the nodes represent exonic blocks and the edges connect blocks that follow in a transcript. Edges can either be splice junctions or internal, within an exon, if there is an alternative splice site. In this example, there are 3 transcripts, all starting at and ending in the same exon. However, transcript 1 lacks the second exon, hence there is an edge from X1 representing the first exon to X4 representing the third exon. Further, there are 2 versions of the second exon, that's why it's split up into 2 nodes, X2 and X3. For transcript 2, X2 is connected to X5, while transcript 3 also includes X3. So X2 is connected to X3, and then X3 is connected to X5. To define alternative splicing events, I searched this graph for what I call splice bubbles. This is a substructure in a graph starting at a bifurcation node and then rejoining all paths at an end node. This provides a binary decomposition of the involved paths, where the 2 sets of paths do not share any intermediate nodes. The 2 sets correspond to the 2 alternatives of the splicing event. Depending on the involved edges, we can classify the event type as exon skipping, intron retention, alternative 3' or 5' splice site, and mutually exclusive exons. All events can be quantified by the % spliced index, which is the number of reads supporting one alternative over the total number of reads. The example here represents the alternative 5' site of the second exon. The purple path corresponds to transcript 2 and the orange path to the version from transcript 3. Here's another example in the same graph representing the exon skipping of the second last exon. The purple path again represents transcript 2 and the orange path the other two transcripts. So the percent spliced index in this case would be the number of reads in transcript 1 and transcript 2 together over the number of reads in all three transcripts. The last bubble in this graph is a little bit more complex, representing the mutually exclusive exons 2 and 3, where transcript 2 and transcript 3 corresponds to the orange paths and transcript 1 to the purple paths. Now, since we can quantify the individual events, we can relate different samples with respect to alternative splicing. For this application, I have a nice use case with sample from leukemia patients. A common mutation in chronic lymphoid leukemia is in SL3B1, which is involved in the assembly of the spliceosome at the 3' splice site. When looking at all alternative spliced events, the principal component analysis does not separate the mutated from the unmutated patients. However, looking only at 3' alternative splicing, patients can be clearly separated, highlighting the benefits of this classification of the events. Of course, the next question is what specific events are different between the groups of samples. To address this, I implemented a statistical test based on likelihood ratio tests with a better binomial model. Like the negative binomial model used in RNA sequencing, this model can handle the variability within a group of sample. Applied to the leukemia samples, we find, as expected, a strong enrichment of 3' alternative splicing, with several examples known from literature, such as Septin8 and MAP3K7. Of course, there are many more methods implemented in IsoTools, as described in our preprint on bioRxiv. If you want to try the package, it's open source, available on GitHub, and in Python package index, so it's very easy to install with the pip command. There's also extensive documentation and tutorials with use cases on Read the Docs. We also contributed to the LRTS challenge, which aims at benchmarking the available long-read transcriptome protocols and methods, and initial results look promising for us. If you're also coming to the ECCB in Barcelona this year, we plan a hands-on tutorial session on IsoTools and the other long-read tools there. Finally, if you have any questions or request support with the package, or if you want to provide feedback, you can use the GitHub functionality or write me an email or visit my poster in the poster session. Last, special thanks to everybody involved in the project, from my department here at the MPI in Berlin, from Michael Schweiger Group in Cologne, which sequenced the leukemia samples. And thank you for your interest in IsoTools.